So good morning. Thanks to the LGC for inviting me to give me this chance to share some of our recent work with you. So I'm presenting on the topic on plasma DNA as a platform technology for cancer detection. For the last few years, there's been a lot of interest in the use of cell-free DNA in plasma for a variety of molecular diagnostics. This field has generally been called liquid biopsies. So we started this field by looking at a model system of nasopharyngeal cancer, or NPC for short, which is a head and neck cancer which is very common in this part of the world. And one reason why we decided to start with NPC is because for this liquid biopsy method to work, we need to have a DNA species which is specifically associated with cancer. And for NPC, we have that in form of a virus called Epstein-Barr virus, or EBV. So about 20 years ago, I hypothesized that maybe in the course of its biology, the tumor will release the EBV DNA in the bloodstream. So when we did experiment, we find that actually in the plasma of NPC patient is really stuffed full of the EBV DNA. On the other hand, if individual without NPC, then most of them are negative. And then more recently, we wonder whether this technology can be used for the early detection of this cancer. Now, we think it's worth doing because, like many cancers, the earlier you detect NPC, the better is the prognosis. Now, for example, here, you look at the survival curves. If you detect NPC in the stage 1, which is a top curve, then actually generally in the long term, over 90% people will survive. But however, if you detect in stage 4, then over the long term, some 40% people will die. But unfortunately, in Hong Kong, some 76% NPC is only discovered late in stage 3 and stage 4. So we would like to do something about that. So a few years ago, we started a trial in which we actually want to recruit 20,000 subjects in Hong Kong, and then in the highest risk group, who are males between age of 40 to 60, we'd like to see whether this liquid biopsy method can result in the early detection of this cancer. So basically, we recruit the individuals, and everybody is subjected to a plasma DNA test. If the test is negative, then the patient is classified as negative. But on the other hand, if the patient is positive, then we wait 4 weeks and we test again. And then if the individual is positive in both cases, then we call them persistently positive. And then we refer to an endoscopy and MRI. And then we treat any cancer that we see. But overall, we have recruited just over 20,000 subjects, in which 5.5% of them are positive in the first test. And 1.5% of them are positive in the second test. In the end, 34 cases of NPCs are found. So the positive predictive value is 11%. Now this number may sound low to some of you, but it's actually already very high, because for actually most of the cancer screening tool which is in clinical use now, we're talking about positive predictive value of 2 to 3%. Now these numbers are very interesting because NPC, the incidence in Hong Kong is about 35 per 100,000. That means if you screen 20,000 people, you would expect only to get 7 cases of cancer. But here, we're getting 34. So we're hypothesizing that maybe we're actually revealing cancer which would naturally present over the next 4 to 5 years. So if that's the case, then the cancer that we uncovered should be earlier. And indeed it is. Now, for example, without screening, then most of the cases are in stage 3 and stage 4, which is the orange and red bars. But however, with screening, you can see that we now shift to so that 70% of cases become stage 1 and stage 2. Now, if they're so early, then maybe that would translate to a survival benefit. And it is. For example, you can see that of the 34 cases we identified in the top line, only 1 actually died. The rest are essentially cured. But whereas in the bottom line is without screening, that over long term, 40% of them will die. So now the chance of dying is actually reduced 10 times. So this work was published in the New England Journal of Medicine. And then we're now very eager to push this out in the clinic because we think it's going to save life. But however, we have a logistic problem. And the problem is that currently the protocol requires the test on 2 occasions. And you know it's difficult to roll out this sort of test if you have to test on 2 occasions, almost like the COVID vaccine when you have to vaccinate in different times. So we are thinking, is it possible that we can compress the 2 tests into just 1 test? So the question is, how do you do that? So currently, we have 3 groups of individuals. In the top group, we have people with the cancer who are positive on both occasions. And then in the middle here is an individual without cancer, but somehow they are persistently positive. They're also positive in both occasions. In the bottom there are individuals without cancer, and they are transiently positive. Positive in the first time, but negative in the second time. But all of them have EBV DNA in the blood. So we're thinking maybe we can get rid of the second test if somehow I can demonstrate that the EBV DNA in circulation are different between a cancer and non-cancer group. So how do you do that? So we're thinking, well, maybe first we'll capture the EBV DNA in plasma. And then we ask, are there quantitative difference between the cancer and non-cancer group? And indeed, we find, for example, here, as you move from a transiently positive to persistent to the cancer patient, there's a gradual increase in the concentration of this DNA. And we can only see this by sequencing, because, for example, a New England Journal paper, we use PCR, and using PCR, we cannot see the difference. So precision of sequencing is essential here. The next question we ask is, are there some biophysical differences between a cancer viral DNA and a non-cancer viral DNA? And then we decide to measure the size of those molecules. And then here, for example, the black line is the line of the patient's own DNA, and the red line is a virus DNA in patient with cancer. So you can see somehow the virus DNA in circulation is just a little bit shorter than the patient's own DNA, non-tumorous DNA. The virus DNA is from a cancer. But on the other hand, in individual without cancer on the right-hand side, then the virus DNA is a little bit different. Because as you may know, 99% of us are actually chronic carrier of EBV. So we also have some virus. But the nature of that virus in the blood is a bit different from a cancer. So then we decide to actually differentiate 2 graphs. Of course, you can measure all sorts of things, but we decide to actually measure this area in the middle here from between 80 to 110 base pair, and we work out a size ratio. And interesting, by using a size ratio, I can differentiate the 3 groups of individuals from transient positive to persistent to the cancer. So next, we then decide to draw some diagnostic cutoff. So in both cases, so we have concentration and we have size. So in concentration, we decide to start with the NPC group on the right-hand side, and then we draw 3 standard deviation away from it. And this is the diagnostic cutoff. And then we do the same thing for the size, so NPC group and 3 SD away, and this cutoff. And by using this, basically, we can cluster the NPC patients together. So on the x-axis is the size, and the y-axis is the concentration. And actually, so by combining this, the diagnostic power is pretty good. For example, the ROC curve, which is a measurement of how good it is, then it's actually 0.97, which is very close to the 1, which is perfect. When you just use PCR alone, it's 0.75. You do counts alone, it's 0.93. You do the size alone, it's 0.92. So basically, by using this technology, we now have a way to detect this cancer with a sensitivity of over 97%, specificity of over 99%, and positive predictive value is 19.5%, which is double of what we publish. And furthermore, only 1 test is enough. And then we wonder, well, can we push this even better? So we decide now, how about if we combine epigenetics, so the modification of virus genome. And of course, the best known modification is DNA methylation. So we decide to look at different types of these EBV-associated diseases, like infectious mononucleosis, certain lymphomas, and NPC. And then we profile the methylation status. And interestingly, we actually find the methylation status are different. For example, here we do some hierarchical clustering, and you can see if infectious mononucleosis is here, the lymphoma is here, and the NPC is here. And then we basically now convert that observation into biomarkers in blood. And once again, you can see with these epigenetic markers, I can differentiate people with transient positive, the persistent positive, and the cancer. And then you can add this methylation to the count and to the size. And this time, we now have a positive predictive value of 36%. So in other words, within a space of 2 years, we've increased it from 11% 2 tests all the way to 36% 1 test. You can see a gradual improvement. This is count only, size only, count and size, PCR and the methylation, etc. And then we actually have formed a company called T12, meaning we want to give people a second chance in health. And actually, we have now launched this test. It's called Prophecy. So it's now available in Hong Kong, and the ramp-up rate is pretty good. And we're now going to push into the Greater Bay Area. But NPC is only the first step. So we actually want to do it for other cancer as well. But of course, not all cancer is associated with virus. So it's a bit challenging. So normally, if you talk about detecting tumor DNA using sequencing, then people will talk about mutations, so change in the DNA sequences. And typically, a tumor genome will have several thousands to maybe tens of thousands of mutations. Now, while it's quite doable if you know the patient has cancer, it's not so doable if you do not. If a patient is a previously healthy individual who's just screening for cancer, then it's a bit difficult. So we're thinking, well, maybe the mutation is not so good. So we decided to move from mutations into the epigenetics, which is once again a modification of DNA in which the sequence doesn't change and only have some modifications. The reason why this is advantageous is because whereas mutation, you're talking about several thousands to tens of thousands, in the epigenetics, we're talking about 28 million in our genome. So the search space is much larger. So we've developed an epigenetic method in which we can differentiate multiple types of cancer. So that we published in 2013. And second, of course, if you tell the individual that you have cancer, then the patient is going to ask you, where is my cancer? So as it turns out, different organs in our body have got different epigenetic profile. So we have now also developed a method called plasma DNA tissue mapping to actually locate where the cancer is, which we did in 2015. So this combination of technologies was actually chosen by MIT Tech Review as one of the top 10 breakthroughs of that year. So that's one way. But however, we still have one Achilles heel. And Achilles heel is conventionally you sequence to look at genetics. But epigenetics is different because the sequencer don't tell you epigenetic information. So usually you have to do something called bisulfite conversion, which is a very nasty chemical that if the C is methylated, that after conversion remains as C. But if it doesn't have methylation, they change into uracil, which on sequence will look like a T. But the problem is that this nasty conversion may kill 90% of your DNA. So I've been thinking, well, is it possible that I could develop something which does not require this bisulfite conversion? And then we decided, well, maybe I should look at some other newer type of sequencer. The reason is because the second generation sequencer I use, like from Illumina, requires a PCR step. But when you PCR, the epigenetic information is gone. But however, with a newer third generation sequencer, I don't need to do PCR. So maybe during the sequencing, the epigenetic information is already there somehow, but only people don't know they're there. So I want to draw an analogy. It's just like you're driving your car, and suddenly in front of you, there's an obstacle. So when you see an obstacle, you slow down the car, and then you drive around the obstacle, and after that, you speed out. So even without I seeing the obstacle itself, but looking at your trajectory, I can predict there may be an obstacle there. So in other words, what we're hypothesizing is that if there is some DNA methylation changes, maybe the polymerase, when it's doing the sequencing, will have some funny kinetics. So for example, like the polymerase will come and copy the bases and emit some pulses. So maybe the duration of pulses will be different, or maybe the duration between pulses is different, and the sequence context is different. So we call this the holistic kinetic model, the HK model, also named after Hong Kong. So basically what we do is that we want to sequence this region. Let's say that there is some sort of epigenetic change there. So we look at the sequence context, and we measure those durations, and then make a table like this. And because DNA is double-stranded, so when the polymerase comes to the other side, we do the same thing, and we copy that. And then we combine the two tables together. Now, when you look at the table, this looks like some sort of graphics to me, like paintings or something. So we wonder whether some of the AI technologies which are developed for looking at two-dimensional structures can be trained to recognize a methylation status. So we use something called convolutional neural network, CNN for short. So we train such a network by using known information, and then we validate it. So to train it, we basically treat some DNA with a methylase, so that is a methylated control. And then we also use an unmethylated control, which is I just PCR it, because when you PCR, you lose the methylation information. And interesting, when we do that, we find actually this is very accurate. The area under curve range from 0.94 to 0.97, and this is a training. And then the validation data set is basically exactly the same. And then we decided to test this a bit more by looking at cellular samples. So we look at placenta, we look at some liver cancer tissues, we look at some non-tumorous liver tissues, we look at blood cells, we look at some cell lines. And the x-axis is a gold standard, bisulfite sequencing. The y-axis is an HK model. So you can see the correlation is very high, 0.99. And then we do a deep dive. So this is a circles plot in which the genome is plotted like a circle. The inner ring is a HK model. The outer ring is a gold standard. And the color is a methylation level. You can basically see that its correlation is extremely high. It actually ranges from 0.85 all the way to 0.98. So basically it works. And we think this method is very direct and intuitive. The reason is because, let's say this genomic region, this bit is methylated, which is the black dot. This bit is unmethylated, which is a white dot. If you use bisulfite sequencing on an Illumina machine, you always only have some very short reads. You never have a read which will span across the whole genome. But here, with the HK model, you can see that I can have a continuous read across this whole region. So you can form a haplotype. You can allow you to tell the interrelationship of methylation sites. And also another thing is this. So let's say you use bisulfite conversion, you see a T there. Without looking at reference human genome, you wouldn't know whether reference human genome has a T there or whether it's originally an unmethylated C which got converted into a T. But however, if I use HK model, I can look here. I know exactly this is an unmethylated C, this is a methylated C, even without looking at reference human genome. So this work was published earlier last year. So methylation is one way. Now, another way is this. From an NPC story, I told you about the power of this size analysis. And we wonder what happened with other cancer, the size of DNA in plasma. Now, for example, if you look at you and I in normal individual in plasma, our plasma DNA has a peak size of 166 base pair, which is a nucleosome size. But interesting, we look at plasma from cancer patient, depends on how much cancer DNA they've got. The more cancer DNA they've got, the smaller is the size. It means the cancer DNA is a little bit shorter. And this shortening is actually reminiscent of what we see in the virus DNA in the NPC. So it seems like this is a general way to do this, the size profiling in plasma. And then we thought, well, if I want to commercialize this or know this better, I need to know biology even more. So the question is, what is the mechanism whereby this cutting of fragmentation is generated? So we think the fragmentation may be generated because of some enzyme, like nuclease. Now, if that's the case, then one way to answer this is to knock out nuclease gene in certain mouse model. Now, for example, you take some blood from a wild-type mouse, control mouse, and then you make an Illumina sequencing library from it. Then you see a band around 300 base pair. And this is a nucleosome size, 160, plus the 140 base pair from an Illumina adapter. So this is what happens. And then we wonder, well, with nuclease, then one famous nuclease is DNase 1. So what happens if I knock out DNase 1 gene in a mouse? But strangely, nothing happens. As a matter of fact, we feel so strange about this that we actually publish this as a negative report, just to alert people about this. But if it's not DNase 1, what else it is? So DNase 1 normally like to cut pure DNA. And then we think, well, the DNA in our blood is not pure DNA. They are DNA complex with histones. They are chromatin. So the question is, which enzyme like to cut chromatin? And the answer is an enzyme called DNase 1-like-3. Some people call that DNase gamma. And interestingly, when I knock out DNase 1-like-3 gene, you start to see some longer molecules appearing. And if I do double knockout, it's the same thing. So here I'll show you, when you knock out DNase 1-like-3 gene in red, there's some longer molecules coming. And then we wonder, well, when this DNase 193 cuts the DNA, does it recognize some sequence particularly? So we decided to take a look at this N sequence I call a N motif. Now, if, for example, you look at a 4 nucleotide N motif, theoretically, there should be 4 to the power 4 possibility, which is 256. So at random, each of those should be present at a ratio of 0.39%. But strangely, when I look at wild-type mice, the top motif are actually much higher than that. It's like 1.5%. But when I knock out DNase 193 gene, the motif frequency changes completely. Now, if you're very observant, you see actually the first 6 of those guys all start with CC. So maybe this enzyme like to cut CC. So basically, we group them together and found in wild-type animal, 7.5% all plasma DNA start with CC. You knock out DNase 1 gene, nothing happens. You knock out this DNase 193 gene, then motif frequency is half. So we think this work is interesting because it's the first demonstration of a link between nucleus biology and circulating DNA. And of course, we also show you there might be some change in physiology or pathology, so like gene knockout, et cetera. But we only have the data in mouse. So what happened in human beings? So in collaboration with Stefano Volpi from Italy, Linda Haraki from the Sick Kid Hospital in Canada, and Boris Rissis from New York, we actually managed to find some human subjects in which we have homozygous mutation of DNase 193 gene. And those people actually have familial systemic lupus. And interesting, when you look at the blood, the result is exactly the same in mice. So in the healthy individuals, it's about 10% is occupied by CC motif. But if the individual have the mutations, then it's become like 5%. And furthermore, if you look at those motifs, then you can see that those individuals with homozygous mutation are really different from healthy individuals and people with sporadic SLE, et cetera. And then we wonder, okay, can we use this motif as a new type of biomarkers? So we take a look at cancer. And interestingly, we find that in most cancer, that's aberrant expression of DNase 193 gene. In most cancer we see, actually is reduction in this. Now, for example, like this is liver cancer. Now, so if DNase 193 like to create a CC motif, if you have reduction of it, that means that CC motif should be reduced in the plasma of cancer patient. And indeed, we find that is the case. You can see this CC motif in liver cancer patient is reduced. And then you can see that just by looking at the motif at the end, I can differentiate people with and without cancer. Now, so we're thinking that, wow, if that's the case, then maybe there will be an early cancer detection system, and maybe we can save lives. So with the experience we gained from my previous work in prenatal testing and the NPC I told you, then myself and 2 of my colleagues decided to actually start another company. So this company is called Serena, which stands for Circulating Nucleic Acid. It's basically a company that allows to detect cancer at an early stage and to allow us to do something. But interestingly, at the same time that we founded Serena, in the Silicon Valley, Illumis just founded another company called Grail. And you can see the mission statement Grail is exactly the same, right? So you can see Serena and Grail basically going towards the same direction. And in 2017, we decided to merge together. So this picture was taken at the merger party, which happens in the NBA Finals. So that year was in San Francisco. So this is the first CEO of Grail. He's Jeff Huber. He's a guy who brings you Google Map. So now, after several years of work, we have now actually launched this Gallery test, which is based on the technology I described to you just now. So the multi-cancer detection using methylation marker and the localization using tissue-specific methylation. And actually, just now, actually, Time Magazine has grouped this as one of the top inventions this year. So we're very encouraged by this. So finally, in summary, hope I've convinced you that plasma nucleic acid is a treasure trove for molecular diagnostics. And today we talk about some new fields called fragmentomics, which is the fragmentation patterns of plasma. And we talk about this intriguing link to nucleus biology. We talk about this new technology of HK model, direct methylation analysis. And then we talk about this NPC screening, which is potentially life-saving. And finally, we talk about the first generation multi-cancer early detection test. And this is a recent review that we published in Science. So those of you who are interested can read this. And finally, I'd like to thank individuals from my group for generating data which I present to you. And of course, without TRS, we won't be here. So thank you very much.
So thank you, Professor Low. If you have any question for Professor Low, please raise your hand to let us know.
Yes, Tech.
Nannis, you're a genius. I really just, my hat's off to you. But recently there's a new group of DNA that is called extrachromosomal DNA. And they're found in mostly GBM, ovarian cancer, head and neck cancer. And these are about 1 million base pairs, and they get rolled off the chromosomes, and they basically carry amplified MDM2, EGFR, MYC, et cetera, et cetera. I wonder if those DNA copies could be exploited to get at diagnosis and prediction of treatments.
Actually, thank you, Tech, for your kind words. So we're recently also looking at ECC DNA. I suppose that is what you mean, right? Extra chromosomal circular DNA. And they actually definitely is detectable in plasma. And they are also surprisingly stable because a lot of the enzymes we talk about are exonuclease. And those guys, if they're circular, they're actually very stable. And also they have a very intriguing size profile as well. It's extremely precise size profile. You know, like just now I show you there's a range, but in the ECC DNA in plasma, there's exactly like 202 base pair, 420 base pair. It's extremely sharp. But this is early day. We've done a bit of work showing that in cancer you can see that, and in fetus you can see that. But the significance and the diagnostic applications, I think we need more work. But they are rarer than the linear DNA in plasma.
Yeah, it probably depends on, I think in GBM you may find a lot more.
Okay.
Yeah, and the final thing is that there is a difference between ecDNA. Some are circular and some are linear. And different cancers have different forms. So it's something that may be worth looking into.
Yeah, thank you. So that's why in the science article, the last bit, we say topology of that. So that is a topological thing, which I think is a new an area we definitely need to look into. So thank you. Yes.
Professor Low, that was very impressive. Sorry, that was very impressive. So I just have a quick question. So you mentioned the methylation changes versus the size profiling. Do you have a sense which one comes first in cancer progression? Is there any temporal differences between these two abnormalities?
I got a feeling the basic mechanism is the nuclease digestion. So basically, whatever is changing your chromatin structure will make it more accessible to the nuclease. And methylation is one of those. And then, of course, you have the histone protein changes and other things. As long as the thing can make it accessible or inaccessible to nuclease, then that will change the size.
Okay, so you think the methylation is not necessarily a regulation of gene expression, for instance, oncogenes and the tumor repressors and things like that?
The methylation, of course, as far as the tumor is concerned, is regulating gene expression to give the tumor cells a survival advantage. But if you are talking about linking it with the size in the plasma, then my explanation that is offered is about the nuclease accessibility.
Thanks.
Thank you.
Thank you, Professor Low. Excellent talk. It's easy for laymen to understand also. So proud that Hong Kong scientists can actually get into the world's arena. One question about your original research on NPC DNA research. on your follow-up, is there any additional patients, whether it is from the transient group or the persistent group, who later develop NPC or other EBV-related malignancies? And also, one question is also, you do it in a single time point, okay, in a high-risk group. Is there any need for additional checking later on?
Thank you. You are very insightful. So the question you asked is exactly what we've been asking. So actually, the question is the transient and the persistent false positive. What is the implication of that? So recently, what we have done is that we rescreened the 20,000 people 4 years later. And interestingly, we find that people who are transient positive have a 5 times risk of NPC 4 years later, and the people with persistent have a 15 times risk. So in other words, those are basically risk factor, almost like cholesterol or high glucose. So those guys will need to be screened more regularly. So if you're negative, maybe I can screen you once every 4 years. If you are transient positive, maybe I'll screen you once every 2 years. If you're persistent positive, maybe I should screen you yearly. Yeah. And of course, in the future, we can think about, is there any drug we can take which can even remove that risk? Yeah. Thank you.
Professor Low, very nice talk. I'm now working in exactly about what you talked about, but I'm very curious about this marker in relation to clinical presentation, because a lot of genetic biomarker you talked about can be very beneficial for early diagnosis, right? But once you have this kind of also quantitatively cut-off value, can that actually already associate with the clinical presentation of different type of cancer? And how, if not, how are you going to manage those people with positive signal?
Okay. Now, so one of the answer to your question is, of course, what I hinted just now. So now as you look at this liquid biopsy, even as a risk thing, because just now everybody look at liquid biopsy say that it is now and present, where you have cancer now. But what I was answering the other audience just now is say that, well, in certain situation, there's a risk to tell you you're at high risk beyond this. And of course, just now I talk about NPC. So the next question is, maybe for other cancer type, it's the same. For example, if I use the tissue localization method and show that in this individual, the liver aberrantly release more DNA in the blood, then I want to know, is this person at a high risk of something else? So I think that will require, another TRS project to look. Yeah, thank you. Yes. There's a gentleman over here. Thank you.
Thank you, Professor Lowe. That was a beautiful talk. You made mention of the fact that 90% of the population are EBV positive. Mm. What is the secret sauce here that allows you to remove that as a confounder for detecting EBV DNA related to the cancer?
Okay, thank you very much for a very insightful question. So basically, 99% of us who are chronic EBV carriers, the EBV is in the rest B cell. So inside the cell persists. So that's why it persists. The cell doesn't die. Whereas in the cell-free DNA story, is because of the tumor cell death, etc., they release in the plasma. So the secret sauce is, if you look at blood cells, then you have a lot of false positive. But look at cell-free plasma, then you get what we have got.
So the sample the quality of the sample must influence the veracity of the test.
Yep, so
the DNA, the cell breaks down into DNA release.
exactly. So that's why for our research project, the blood is taken and go to a lab within 6 hours. But if you do it diagnostically, then there are certain tubes with formaldehyde and other preservative called Strack tube, which will preserve your cells so they don't lyse. Yeah, thank you. Yeah, Lab Chief.
Thank you, Dennis. All of a sudden, I remember something I learned a long time ago. Actually, EBV has sort of population differences, put it very simply. And in Chinese, it's called NPC. But in other populations, can you comment on that and any speculation on that?
Yeah, thank you. So actually, we've been inspired by TAC in various discussions. So actually, there's an interplay between the EBV genotype and also the interplay between us, the genome, like the HLA types. So we have actually, Jackie Lam here in the audience, have actually sequenced that EBV and actually look at the polymorphism EBV. And there's certain strains which are high risk. And then the interplay with our HLA type. And certain HLA types which are predominant, for example, in Guangdong people. Yeah, so thank you. So in Africa, the EB manifestation in cancer is the Burkitt's lymphoma. So we have also done that. I have a collaboration with a group in Cambridge to actually look at Tanzanian children. And definitely this marker is also usable there as well. Yeah, thank you.
I just want to add that's not me, it's Maria Lung's insights.
Thank you.
Other questions? Professor Low for his excellent and very inspiring talk.